Prothrombin activator catalyzes the conversion of prothrombin to thrombin. Thrombin catalyzes the conversion of fibrinogen to fibrin. You should know that. Okay, because that's what you'll be questioned on. So that's a nice little synopsis there. And you don't have to know all that, just the part that starts there. Okay?
Just from the prothrombin.
Yeah, but, and also you should know though, that intrinsic involves PF3 produced by platelets, and that could happen in a test tube. Extrinsic, you need damaged tissue, and that involves thromboplastin, or tissue factor as it's sometimes called. Okay, so I've already said all this. Okay. And so you may have noticed in that real snaky, complicated picture that there were all these clotting factors that have Roman numerals. Okay, and ones you could recognize are fibrinogen and prothrombin, clotting factors 1 and 2. Other ones are given Roman numerals, but some of them are proteins. And there's a very famous one, clotting factor 8. For the proteins, there's a gene for them. And if you're lacking the gene, or if your gene is flawed, turned out that Queen Victoria had a flawed gene for clotting factor 8, and that we'll go through the whole story when we talk about genetics in here. And it's Queen Victoria that gave rise to hemophilia, which only occurs in sons because of the fact that it's an X-linked trait. And we'll get into all that. Interesting stuff. So blood clotting, we talked about it. Hemophilia A, lacking the gene for clotting factor 8. They're not lacking the gene, it just doesn't work. Okay, clots shrink. They get smaller, and you may have noticed this in a clot that on the surface, it'll shrink and get kind of hard after a while. First they're kind of larger and softer. This is more important, although it's important on the surface too, because it helps pull the edges of the wound together. But anyway, they're actually myosin and actin filaments present in this fibrin that cause, so the strands actually contract and make the clot smaller. And so makes it more dense, and that pulls the edges of the wound together. So the clots get smaller. Okay. But also, it becomes appropriate at some point, especially if the clot is on the inside. If the clot is on the inside, you need it to go away at some point after you've healed, correct? And so you start to initiate some chemistry that depolymerizes the polymerase form. At some point, you want to chop the chouchy-train back into its little parts, okay, into its little monomers, and so that if you get rid of those fibrin strands, the whole clot will go away. And you want that to happen with an internal clot, particularly. And we want to depolymerize the fibrin. And there is a substance that'll do that. It's called plasmin. But we don't normally have plasmin present, otherwise the clot could never form. We have the inactive form of plasmin, which is called plasminogen. And that is also present in circulating blood. It's an inactive form of plasmin, and the appropriate time, tissue plasminogen activator is released, and that converts plasminogen to plasmin, and plasmin depolymerizes the clot. Okay? The really cool thing about this chemistry is it sees widespread clinical use in the dissolving of pathological clots, like the clots that cause heart attacks and the clots that cause strokes, okay? 91% of all heart attacks are caused by clots, okay? They're acute events that you don't, they're usually initiated by the buildup of plaque inside an artery, but arteries hardly ever become completely plugged up. That just hardly ever happens. They become narrow or stenotic. At some point, though, they become inflamed because arteries don't like having a whole bunch of plaque in them, and they'll start to become inflamed and release clotting factor, basically tissue factor. Thromboplastin, various clotting agents are released, which cause a clot to form and get stuck in the already narrow lumen of this narrow little artery, and that plugs it up completely, and the tissue downstream will die if you don't get rid of that clot really quick. If you live across the street from a hospital and know what's going on, you could probably get the patient over there fast enough to do thrombolysis on them. And thrombolysis was first done at the Stanford Medical Center, but it's practically, I'm sure they do it in places like Eureka and Spade, not to mention Medford, all over the place. And Klamath Falls, maybe even at the hospital in Crescent City, I don't know. Can't speak to that. And maybe at the one, it'd be interesting to find out if they did that, probably not. What you do is identify where the clot is real quick and put a catheter right next to the clot and drip TPA, tissue plasminogen activator, in right next to the clot, and that'll dissolve the clot. Heart tissue dies really rapidly because it has a huge oxygen requirement. If you do it for a stroke, brain tissue doesn't die so fast, surprisingly enough. And very commonly, you can completely reverse the effects of a stroke if you get the patient to a hospital fast enough and do thrombolysis on them. And, so that's good to get the patient to a hospital in many cases pretty rapidly. Okay. I'd say it's much more useful in the case of strokes than heart attacks. So plasmin depolymerizes fibrin. Okay, process we call thrombolysis, which is used commonly these days. Catheters placed in a vessel next to the clot, and used to be they would use streptokinase, but they found a way to produce human TPA now. So it's almost exclusively TPA, which is made by recombinant DNA synthesis, and that's what they do. It's called thrombolysis. Very cool clinical procedure. Okay, prothrombin time measures whether your blood clots too rapidly or too slowly. If it clots too rapidly, you're a candidate for all kinds of pathologies, embolisms and stuff like that. There, you give an anticoagulant like dicumarol or warfarin or one of those. I also saw a seminar on one of these Friday afternoon seminars, the guys who invented Coumadin, all the Coumadin derivatives. And at that time, they didn't know that it was good for anything, but it was just an interesting chemical reaction. And I doubt they ever got any money out of it, but whoever they worked for certainly did, because it's one of the most widely used pharmaceuticals there is these days, and they discovered this bizarre chemical process to make it. So, okay. So they do prothrombin time all over the place. Here in Brookings, you can get it done. And they'll prescribe, basically you titrate the dose of dicumarol against your prothrombin time until you get a normal prothrombin time. And the thing is though, you gotta eat a pretty constant dose of vitamin K, because dicumarol is a vitamin K inhibitor. And gotta eat the same amount of vitamin K all the time, is what I'm trying to say. When you're titrating your dose of dicrumarol, if you change the amount of vitamin K you eat, you'll have to go in and retitrate your dose, which is kind of a pain. So, there are no systemic coagulants, only anticoagulants. So, okay, and anticoagulants, the Coumarol drugs, dicoumarol, Coumadin, warfarin. Okay, and the dosage has to be titrated against one's vitamin K intake, basically, and you measure, do prothrombin times until you get the right dose. And dicoumarol inhibits an enzyme which recycles vitamin K. And broccoli contains huge amounts of vitamin K. If you're a broccoli lover, you gotta eat the same amount every day. Okay, so now we'll talk about our final topic for the evening, blood groups. Okay, blood cells possess cell markers on their surface. All cells possess cell markers on their surface which help identify those cells for our immune systems, so that our immune systems can know whether or not the cells are self or non-self. To our immune system, there are only two kinds of cells, self cells or non-self cells. If our immune system comes in contact with what it identifies as a non-self cell, it will destroy it. You don't want that to happen to your blood cells. Okay, so when they're doing transfusions, they get all concerned about this because blood cells have a number of these cell markers, which are called antigens, on their surfaces. Okay, so we're going to talk about the A and the B antigen. There are about 50 different blood antigens. These seem to be the most important ones, but there are a whole bunch of other ones. And if you're someone really important, like the president, they'll have your blood cross-matched for all 50 antigens, however many there are, and make sure they have a whole bunch of blood on hand, which exactly matches, all of those blood antigens. So, we need to talk a little bit about the immune response, because blood typing is an immunological assay. It's a test that uses antibodies, okay? And an antibody is like a receptor, only it's a receptor that floats around in blood, okay? And this receptor is capable of plugging onto an antigen as just like any other substrate plugging into a receptor, sort of a key plugging into a lock. And hypothetically, there is only one substrate that will fit an antigen, okay? And antigens are pretty discriminating. Pretty hard to find more than one substrate that will fit into a given antibody. And there are whole catalogs that you can order from, that you can order antibodies to various substrates from a catalog, and they're expensive, and you can have these antibodies on the shelf, and you dissolve a little bit of them in water and use them to test whatever antigens you're interested in. And of course, we're interested in the antigens on the surface of blood cells. And so we're interested in the A and the B antigen. So you would purchase copious quantities of A and B antibodies and have them on the shelf. So when you do blood typing, maybe you've seen these little white ceramic trays that have a bunch of little indentations in them, and they'll put little drops of blood in there, and then they'll drop antibody A in one of them, and antibody B in another one. And if you put the antibody together with the antigen that matches it, you can visually see a reaction. It's called an agglutination reaction. Their precipitate is actually formed. You don't need a microscope or anything to see it. So it's kind of handy. And so if you add A antibody to your blood sample and you don't get an agglutination reaction, then you know you don't have the A antibody. If you add B, if you know you don't have the A antigen on your blood cells, if you add B antibody to your blood cells and you do get an agglutination reaction, then you know that your blood sample does have the B antigen on its surface, and you draw appropriate conclusions. So as usual, I'm jumping the gun. When a foreign antigen enters the body, the immune system produces antibodies which are specific to that antigen. Normally, a person must be exposed to the antigen in order to make antibodies. So I presume they use horses or something like that to make huge amounts of these antibodies. And they use horses to make antibodies to snake venom and all kinds of things, just because horses are very tolerant and they're big and they can make a lot of it for you. And so anyway, wherever they get these antibodies, they're called monoclonal antibodies. You can just get a bottle of just one kind of antibody, and that's all there is in there. And this is evidence that holds up in court. Okay, antibodies are very specific. So if you add an antibody to an antigenic substance and see an agglutination reaction, then you know that antigen was in there. It's ironclad. So, okay, when antibodies react with antigens, an agglutination reaction is observed. Agglutination reactions are discernible to the naked eye and are very specific. So, these are the blood groups that we'll be interested in. We're only talking about two kind of antigens here. Blood type A possesses the A antigen, and only that one. Blood type B possesses the B antigen, and only that one. Type AB possesses both of them, and type O possesses neither of them. Okay? So, here we have blood type A. Okay? And this didn't copy very well, but it was a great color slide at one time. They don't scan very well. We added A antibody to type A blood, and we saw an agglutination reaction. So, what is the conclusion? We have type A blood. Okay? This is how you would prove that you had type A blood. This is how they even came to know that this was type A blood to begin with. Someone had to do this. Okay? They add B antibody to type A blood, nothing happens. Okay? So this is how they do it. I got quiz questions. Which of the following is true if you add if you add A antibody to a blood sample and get an agglutination reaction? And I give you a number of choices, and the answer is that it's type A blood. Okay?
So what happens with an A and B type blood? Do you add both?
Well, we'll go through and look. Okay, here is type AB blood. So we add A antibody to type AB blood, and you get an agglutination reaction. And you add B antibody, and you get an agglutination reaction as well, because there's both the A and the B antigen. That's the definition of type AB blood, is you have both the A and the B antigen. So you'll get an agglutination reaction with both types of antibodies. This is exactly what they do when they do these tests in the lab. Yeah.
What's the positive or negative?
We'll get to that. Has to do with the Rh factor. So here's group type B blood, and for type B blood, you add A antibody and you see nothing. But with type B antibody, you add it to your blood and you get an agglutination reaction, which is what you expected because you already knew it was type B blood. And if you didn't already know it was type B blood, you know it now. Okay? So, and of course type O has neither the A nor the B antibody. So you add A antibody, no reaction. You add B antibody, no reaction. here's the color picture in your book. And antibody B to type A, no reaction, reaction with A antibody. Here we have B antibody to type B, you do get a reaction. But yeah, with type B blood with A antibody, no reaction. Type AB is going to get a reaction with both of them. Okay? And you can see it on my TV here, even if you can't see it. So blood type distribution, for what it's worth, I'll never ask you anything about that. So here's the thing that doesn't jive, and I have no explanation for it, and nor has anyone that I've ever talked to. For some odd, normally you have to have circulating antibodies to some antigen, you have to have been exposed to it, okay, at some time in your life. That's how they test for HIV. They find HIV antibodies. So they know the person has to have been exposed to it. However, for some odd reason, people with type A blood automatically are born with, it would seem, the circulating B antibodies. We are born with antibodies to the antigen we don't have. Okay, how did we get that? You got me. I have no immunology is a hobby of mine. And I have no immunological explanation for that, nor has anyone that I've ever talked to. Okay, it's contrary to the laws of immunology. And I've had colleagues of mine say, well, what if they meet? You know, well, I think they test, a lot of times they test newborns. And it's pretty hard for these newborns to eat a bunch of meat, you know, before they got tested. The meat, you know, could contain the other antigen. But who knows? So anyway, so if you're type A blood, you'll have B antibody. If you're type B blood, you'll have A antibody. What if you're type AB blood? You'll have neither of them. You're born with the antibody to the antigen. Let's see, you're born with the antibody to the antigen you don't have. So maybe, let's see. Yeah, if you're type AB, you can't be born with the A or the B antibody because they would attack your blood. See? So you'd be born with neither of them. Okay? And if you're type O blood, you have neither antigen, so you have both antibodies. So type AB is a universal donor in one sense. But let's go through
Well,
this.
I thought type O was the universal donor, type AB is the.
Yeah, let's go through it. So here's what I just said. If you have type A, you'll have the B antibody. If you have type B, you'll have the A antibody. If you have type A, you'll have neither antibody. And if you have type O, you'll have both antibodies. Okay? So if you have no antibodies, you'll be a universal donor because you'll have no antibodies to attack that other person's. That's one way of looking at it. Okay? To attack that other person's antigens. So, what blood can you be received? O is the universal donor. And these types, depends on what, you know, I'm starting not to like this universal recipient, universal donor terminology because
But can't
it
O
depends on how you
only
define
get
it. Yeah, it depends on whether you define it with regard to the antigen or the antibody. Okay? So if you have type O, you have no antigens, so that would be very favorable to the recipient because you have no antigens on your blood cells. so that'd be good, except you have both antibodies. So it depends on kind of how you look at it, it starts to break down, that recipient and donor. But let's just say that type O is a universal donor because there are no antibodies, no antigens on the surface of the cells. Okay, but there's all kinds of antibodies. And so Camille,
So isn't O only supposed to receive O though? It can't receive any other?
well, O has both antibodies, and so it won't do well with, it's certainly not a universal recipient. So if you give, you sort of need to take it on a case-by-case basis. If you give type A to type O, it'll have antibodies to that. It has antibodies to all other three types. Because it's got both the A and the B antibody. So it's got antibodies to all the other three types.
But you can get the AB type, right, because it doesn't have any,
So it depends on how you look at it. Type O is pretty much, you want to give type O to. Okay? So it depends on whether you look at it if you're looking at the antigens or looking at the antibodies. Okay? So a person with type O on their blood cells has no antigens, but they do have both kinds of antibodies. So they will attack anything but type O. So this one has it has both antigens, but no antibodies. So that makes it advantageous in one sense. It has no antibodies, but it's got both antigens. So the best thing to do is to match, get the closest match you can, give type A to type A, and, to be safest. But it's not uncommon to see untoward reactions when you do transfusions, because you gotta remember all these other antigens that we're not even talking about. And a lot of times you've gotta do the best you can, but, certainly be careful who I gave type A, AB to, because you got both antigens. So finally, what's all this A negative, AB negative, and B positive, and all that kind of stuff? We're talking about the Rh factor, which is another important blood cell antigen. And the importance of this lies in the fact that, and of course you can buy your little bottles of antibodies for the Rh antigens that everyone has on their shelf who does this testing. Along with the A and the B antigen, they have the Rh antigen. And this is particularly pertinent where you have a couple that are gonna have a baby. Because it's possible for the mother not to have the Rh antigen. Rh comes from, they first found this antigen in a rhesus, on blood cells of a rhesus monkey. And that's how they came, so rhesus starts with Rh, and so somehow it got, they later came to call it the Rho antigen, for the Greek letter Rho, R-H-O. And so anyway, suppose the mother is Rh, her blood cells don't have this Rh antigen, or the Rho antigen. Her immune system has never seen it. During our childhood, our immune system takes inventory of all of our antigens and learns to identify all of our antigens as self. Okay? After that time, any antigen that comes along isn't recognized by our immune system as self, and if you're not self, then you're non-self, and non-self means you get killed. Okay? And so if the mother has never seen the Rh antigen because she doesn't have it, then her immune system would regard it, if it were to ever see it, as non-self and mount an immune response against it. Okay? So suppose that this woman marries a husband who does have the gene for the Rh. This woman doesn't have the gene for the Rho antigen. Suppose she marries a husband who does. So his blood cells will possess the Rh antigen, which is easy to test for. You're just doing a simple agglutination test. And so there there's a chance, there's either a 50% chance, or when we talk about genetics, I'll talk more about this, but there's at least a 50% chance that the child will possess on his or her blood cells the Rho antigen. And so fetal and maternal blood do not mix. There's no, blood cells cannot cross the placenta. However, at the time of birth, there is some mixing of fetal and maternal blood. And so at that time, the mother's immune system for the first time comes in contact with fetal blood, which possesses this non-self antigen, and the mother mounts an immune response against that blood, and forevermore, in perpetuity, she will then possess circulating antibodies to that Rh antigen. So the next time she becomes pregnant, antibodies do traverse the placenta. And the next time she gets pregnant, she would mount an immune response against the fetus, and it would never stand a chance. Because it would never even become a fetus, it would never even become much of an embryo. But at some point, when the blood cells start to express antigens, it would be, killed. And so what they do, this is just what I described to you about the immune response. I don't think we have to go through that again. But, the risky situation is if the father is Rh positive and the mother's Rh negative. Is it the first child that's at risk?
No.
No. Okay, good response. It's only subsequent children that are at risk. And so if the child possesses the Rh antigen, the mother will mount an immune response. Okay? And so you don't want that to happen. If the mother possesses the Rh antigen, she'll regard the baby's Rh antigen as self. So everything's great. It's only when the mother's never seen it before. So we have, what we do is at the first time that you suspect that there was mixing of fetal and maternal blood, you administer this drug, which consists of antibodies. It's called RhoGAM.